double-sided, one side of the page, or both sides of the page?
Just one side. And if I need a magnifying glass to read it, it doesn't go. Don't do that. Yeah.
Do you give us the page that has lines on it? Or do we?
I'm not going to give you lined paper. No, sorry. Do you prefer lined
That's not a
paper?
crazy question, is it?
I wouldn't say it's crazy. No, I just, no one's ever asked that before, but I'm not going to do that.
First one.
Yeah. Anything else? That's why we put cases on our phones. Okay, anything else? Okay, last thing we were talking about, blood flow in general. Remember, blood flow through our bodies is based on pressure gradients. You have to have a pressure gradient if blood's going to flow from one place to another. It flows from high pressure to low pressure. We saw that if you sort of map the blood pressures out throughout the cardiovascular system, arteries, where blood's leaving the heart, have the highest pressure. Veins, where blood is entering back into the heart, you have the lowest pressure. So blood flows from arteries through the capillaries to the veins. You're going to get a drop in blood pressure all the way through. You have to, or else blood's not going to flow through. Resistance to blood flow. So the more resistance you have, the less flow you get. And we talked about different ways that the resistance can be influenced, like the length of the vessels, the diameter or radius of the vessels, and the viscosity of the blood flowing through it. All those can contribute to the resistance to flow. And then we started talking about flow velocity. So I want to go back over that because I know that's a little confusing. Lots of units and things to keep track of there. So let's try and go through that again. Make sure that's clear in everybody's mind before we move on. Questions before we get back into flow velocity? Yes?
Will you just repeat the resistance one? The more resistance you have, the...
Less blood flow you have. Anything else? All right. So let's talk about flow velocity. So if you increase the radius of a blood vessel, it gets wider, then the flow velocity through that vessel drops. And this is assuming that you're working with a closed system. You've got a set volume per time of blood coming in, and that same volume per time is coming out the other end. So somewhere along the line, we're not adding any blood and we're not removing any blood. What goes in must come out. And as you go through this section, if you change the radius of the blood vessel, it gets wider, larger radius, then the velocity at that section is going to slow down. If it gets skinnier, smaller radius, then the velocity is going to speed up. And that's the idea here. So let's go ahead and try and show why that's the case. So to understand that, we need to make sure we understand what flow by volume is. So that's the volume of blood per time going through the vessel. And we could use the units of centimeters cubed per second. Right? Usually we're going to measure volume in either liters or milliliters. But for this purpose, and I think it'll be really obvious pretty soon, we're going to use centimeters cubed for volume. Flow by velocity, which is different, is going to have different units. This is distance per time, or centimeters per second. Okay, so in our little imaginary system, we're going to have the flow by volume constant throughout the entire length of the vessel. What's going to change is the flow velocity and the radius. So if the flow by volume coming into the system must equal the flow by volume coming out of the system, we can express the flow by volume a different way. We can express that flow by volume as the surface area of the vessel at some area multiplied by the flow velocity at that area equals the surface area somewhere else multiplied by the flow velocity in that somewhere else place. And if we keep track of the units, then it shows us why this can work out. So the units for surface area are centimeters squared. The units for velocity, flow velocity, we have as centimeters per second. Centimeters squared multiplied by centimeters per second gives you centimeters cubed per second. So the units work out. Right? So both sides are going to be equal to each other, right? One represents, area 1 along this vessel. 2 represents some other place in that vessel. So as long as the flow by volume in each place is equal, then we can change everything within there, right? If the surface area goes up, the flow velocity must go down to keep the flow by volume the same. Yes?
Do we have to know these equations the way they are to explain something, or can we just say like, you know, if this goes up, then this will go down, and so forth, and explain it all? So, like,
You mean that top relationship?
Yeah, like.
You just want to memorize that and move on, huh?
Well, I mean, I don't know, do we need to know actual, like, if we have to put input information?
So on an exam, I'm not going to give you numerical values to plug in, but I do want you to know that relationship. Yes, I want you to know that the radius of the blood vessel is inversely proportional to the flow by velocity in a closed system. I want you to know that, but I want you to sort of understand why that's the case. Right, sort of take it that next step. And this is starting to help explain the why. Okay? Yes?
Is there any correlation between flow velocity and, I want to say, varicose veins? You know, like how there's poor flow of blood through the veins, and is that why they increase size?
Well, with the varicose veins, the walls, the integrity of the walls goes down and they sort of stretch out, and the valves stop working. I'm going to talk about the valves. The valves don't capture blood or keep it going unidirectionally, and so that's a little different than this. Yeah. Yes?
So narrowing the surface area, would it be considered adding resistance to?
Okay, so that's a good point. So if you narrow the blood vessel, you make the radius smaller, that's supposed to increase the resistance to flow through there, which would then you would think sort of, you would sort of assume, all right, we're going to have less flow through there. But remember that I already told you that the flow has to be held constant. Okay, so what we're talking about now is, you've got some vessel that changes the radius as you go through. But whatever comes in has got to come out. Right? So when it gets to this larger radius, it's going to slow down. Get a smaller radius, it's going to speed up. Which kind of goes against what we had said back there with the resistance thing. Where that resistance takes effect is if we talk about a different kind of system. So here, there's two options of which way to go. And if you make this part skinnier, then yeah, we may have more resistance there. If this branch wasn't down here, then all that blood would have to still go through there and have to speed up to get through the skinny part. But because there's another option, now that there's more resistance to flow, more blood's going to go this way. And so we're actually going to have less blood coming out here than what came in here. But in this system, the flow by volume that comes in here has to equal these two flow by volumes added together coming out. Right? So this is a different system than that. See that? Okay. Anything else for now? Okay, so here's a figure that kind of shows what we've been talking about. So we've got one of these funny vessels where it's skinny in the beginning and then it gets wider. Flow by volume going in is going to equal the flow by volume coming out. So if we have the smaller surface area, you're going to have to send more blood through per unit time. It's going to have to go through faster. Right? And that's what this wider cylinder is supposed to represent. And when you get to the wide part, this blood is going to slow down. If it stayed going faster, you'd be passing a lot more blood. Well, where is it going to get it from? It's only getting blood at such a rate from upstream. It's not going to suck more blood in. How do you guys feel about this? I heard that sigh. Is anybody getting it? Anybody not getting it?
I have a question. I kind of get it.
Kind of is better than not at all. Okay, what's your question?
You said that blood flow stays constant, or is it constant in the equation?
So we're setting these conditions.
Okay.
We're going to say, hey, we're going to force it to be constant.
Okay, got it.
There's nothing constant
There's
in real life.
Yeah.
Yeah.
Alright.
Anything else? Yeah.
Can you, on the previous slide, just verbalize the last equation again?
Verbalize it.
Yes. Surface area, okay,
So basically read it.
yeah, just read it out loud again.
Just read it, would you? All right, so the surface area of the vessel at position 1 multiplied by the flow velocity at position 1 must equal the surface area at position 2 multiplied by the flow velocity at that position 2.
How would that translate onto your drawing?
Okay, so this horrible drawing over here, let's say this is position 1 and this is position 2. Right, so we've got this sort of unit of blood coming through here. And we've got this unit of blood coming through here at position 2. So we're going to compare surface area at position 1 and surface area at position 2. The velocity at 1 and the velocity at 2.
And then where does the centimeters come in?
So the centimeters is from, the surface area is measured in square centimeters. The velocity is measured in centimeters per second. And then the width here is supposed to represent how fast the velocity, so that the total volume of this cylinder should equal the total volume of that cylinder over a given amount of time. Yes?
How much friction?
How about friction?
Yeah, because any surface you go, this fluid is going to be same, but friction also bring the flow down. Like that can be 100% true because you should put the one percentage there, like it based on that space, you're going to be down based on the friction down and multiply it with this number.
Okay, so let's say this is going the other way, right? So we're going through this nice wide section, all of a sudden we go to skinnier section and there's going to be more friction. Is that what you're suggesting?
Also distance can affect it too.
Distance is what?
Distance too.
Right, because the longer you go along, there's more friction there is. Okay? And so now all of a sudden there's more friction, so less blood can get through here, which means less blood per time is going to leave out this end. But then you can't push in more blood than is coming out because it's got to go somewhere. So then the flow in is going to drop. So again, this whole thing is based off of the volume that blood comes in per unit time must equal the volume that comes out. And yeah, if there's stuff over here that's going to add to the friction and slow things down, the whole system slows down in terms of the volume per time.
Now just according to your formula, because this is SA multiply flow, this should be SF multiply flow multiply V or something as a number of the friction.
So you want friction in here.
Yeah, it's going to be that, make it more accurate.
It's not though. You don't need to consider the friction here to get this relationship. Because I agree that you make it skinnier and you make it longer, there's going to be more friction. But that would slow down, that would decrease the total volume per time that goes through the system. And so as long as it's still equal, even if there's less friction here, it's not going to pass more volume per time than you can pass here, because where would it go? It's got to wait for that blood to get through. But down here, that is important. That's where this is coming in. When we decrease the radius, we're going to add more friction there. It's going to be harder to squirt blood through. And so that does play into effect because now more blood's going to go the other way. There is no other way here, though. That's why we don't have to worry about the friction in this relationship, to understand this relationship between surface area and flow velocity. Okay. Anything else? No. Ready to move on. All right, we talked about that. Why do I want to show you this? There's a heart in the middle there. Okay, so we had talked about this figure before. Right, we use this figure to highlight the two circuits. There's the pulmonary circulation, the systemic circulation. It's all driven by this one heart. And this one pump, this one heart, is really functionally two separate pumps working next to each other. You've got the left side pumping blood out into the systemic circulation, the right side pumping blood into the pulmonary circulation. And those little black arrows tell you the direction that blood flows. And when everything works properly, it always goes in that direction. right? When your heart contracts, right, this sac of blood squeezes down, blood doesn't go in every direction. You don't get blood squirting back into the veins. It's always going to go off to the arteries. So the next thing I want to really take a look at is, why is that the case? You can look at inside the heart specifically, and you can see all these little black arrows showing you the direction that blood flows. Right? Those aren't double-headed arrows. It's only going in that one direction, assuming everything works properly. Right? So when the ventricles contract, why does blood always go into the arteries and not back into the atria? Or from the atria into the veins? And that is because of the heart valves. So we've got these little mechanisms in our hearts to ensure that blood always goes in one direction. It's unidirectional flow. We have 4 valves in our heart. 4 valves. I kind of like to think about these valves as being on either end of the ventricles. So we have valves that control blood flow into the ventricles, and we have valves that control blood flow out of the ventricles. Okay. And whatever we've got on the left side, we've got on the right side. Let's go ahead and look at these valves. So, the first pair that we'll look at, the atrioventricular valves. They tell you where the valves are. They're between the atria and the ventricles. So there's a left AV valve and a right AV valve. Okay? So back to our little drawing. Looking on the right side of the heart, here is the right atrium. Blood leaves the right atrium into the right ventricle. To get there, it has to go past these little flaps. This is the right atrioventricular valve. On the left side, we have something just like that. From the left atrium to the left ventricle, there is the left atrioventricular valve. Be careful with the name, atrioventricular valve. Don't get it confused with an atrioventricular node. Nodes and valves are totally different. Yeah, there can be in similar places, but structurally and functionally, they're totally different. We'll talk about that node a little bit later on. Don't get nodes and valves confused. All right, so how do these things work? They're really pretty cool. These are passive one-way valves. So they only let blood go in one direction, and they're passive. You don't have to use up any energy to close or open these valves. They just open and close when you want them to. So let's take a look at how this works. Let me use my hands as an example first. Let's say that my hands represent the flaps to an AV valve, atrioventricular valve. On top of my hands would be the atrium. Below my hands would be the ventricle. And so if the atria contracts, we get the atrium contracting, it's going to squirt blood out of the atrium through this valve into the ventricle. So we squeeze down the atrium and the valve flaps open, blood squirts through. It doesn't make that noise though. Right? It squirts down into the ventricles fill up, everything's great. All right, now it's the ventricles' turn. Right? And we already know about this separation and contraction between the atria and the ventricles, because we saw that with the lab, the ECG lab, right? With the P wave followed by the QRS complex. Sound familiar? Didn't forget that already, did you? All right, so now the ventricles contract. The atria are done with their job, they're done squeezing. The ventricles squeeze down their contents. And all that blood now is going to want to squirt out into its associated artery. But why doesn't it go back up into the atrium? Because remember, our flaps were just open. All that back pressure pushes on these flaps and seals them. It seals those flaps together, so now blood can't squirt through back up into the atrium. So these valves work off pressure gradients. When the pressure is higher in the atrium than the ventricles, the valves are open. When the pressure is higher in the ventricles, the valves get squeezed shut. And if there's a little leak, then there's a murmur. We're going to assume there's no leak right now. Okay? All right, so passive one-way valves. There aren't little muscles contracting to open or close those valves. It's just based off of pressure gradients. Okay? All right, now we go ahead and look at the picture here. So here we've got an atrium and a ventricle. We actually have these little flaps that make up the AV valve. And so when the pressure is higher in the atrium, the flaps are open, blood squirts through into the ventricles. When the ventricles contract, the pressure down the ventricles is higher than the pressure in the atria. that back pressure pushes up on those flaps and they seal shut. So blood does not sneak back up into the atrium. Instead, it has to work its way out into the arteries. So those are the AV valves. Questions about that? All right. The other set of two valves are the semilunar valves. So we have two of these, one on the right side, one on the left side. And each semilunar valve has its own name, and it's referencing where blood is going. So the aortic semilunar valve is a valve between the left ventricle and the aorta. The aorta is that first main artery in the systemic circulation. It's our largest artery, the aorta. We also have the pulmonary semilunar valve. And the pulmonary valve is between the right ventricle and the pulmonary trunk. The pulmonary trunk is that first artery in the beginning of the pulmonary circuit.
Can you repeat where the aortic is?
The aortic valve is between the left ventricle and the aorta. So these valves control blood flow, or blood leaving the ventricles, where the AV valves, or atrioventricular valves, control blood flow into the ventricles. I mentioned in the beginning that they're on sort of either end of the ventricles. Let's go ahead and take a look.
You said the aortic valve is the largest?
The aorta is your largest artery, and the aortic valve controls blood flow into that vessel.
But is that the largest valve?
I don't know that it's bigger than the pulmonary valve. Well, I'm not sure. I don't know. What's that?
It looks large.
Another picture was different.
Well, in this picture they look pretty similar, right? These flaps are thicker, but the diameter here doesn't look much different. Plus that's an artistic interpretation. It should be taken as such. Right? Alright, so let's look at these semilunar valves. So here we have the right ventricle. When blood leaves, it goes past these little flaps into the pulmonary trunk on its way to the two lungs. When the left ventricle contracts, blood goes through these flaps, through the aortic valve, into the aorta, out to the systemic circulation. These are also passive one-way valves. Right, so here's a little drawing showing when the ventricles contract, the flaps open, blood squirts out into those arteries. When the ventricles stop contracting, the pressure drops. The pressure stays high in the arteries. That high pressure pushes back on those flaps and keeps those flaps shut so blood doesn't leak back into the ventricles. Why is it important that we maintain a high blood pressure in those arteries?
Cluster gradients because of anywhere else?
Yeah, if you're going to have blood flow from your arteries towards your veins, you need to have a high pressure there. So you keep a high pressure in your arteries throughout the cycle. Right? When blood is flowing through your body, your blood vessels, it doesn't flow and then stop and then flow and stop. Right? There's a pulse to it. We'll talk about that pulse a little bit later on. But it does continue to flow all the way through. All right, how do you guys feel about valves? Questions about that? Okay. All right, so now we know, we got a sense for the direction blood flows through the heart. We know why it goes in that direction and not backwards. It's all driven off the pressure gradients in these valves. Let's go ahead and start talking about how we're going to get that heart to actually contract. So, cardiac excitation and contraction. We've got to excite the heart, and then it has to contract. All right, number one, we generate an action potential in the heart. We're going to talk about where that comes from in a moment, but for now, let's assume you have this action potential that starts, and that action potential has to spread throughout the heart. We're going to talk in a little more detail about the path that it takes through the heart, but for now, just realize it's going to spread over all the chambers, so that all of the cells in the heart get the message, it's time to contract. Okay, so here we have, so here's one of those notes I talked about. right? The signal is going to start where these nodes are, and that electric signal, noted in yellow arrows here, will spread through the different cells, telling all the cells, hey, it's time to contract. How do they get there? They can be passed through these gap junctions. So each cardiac muscle cell does not have to release neurotransmitters that diffuse over and then stimulate the next cell. They all act as one cell together, functionally, right? Because these electric signals travel right through those gap junctions from one cell to the next. So the whole thing ends up depolarizing together. All right, so the action potential spreads. When it gets to these contractile cells, it triggers calcium ions to come into the cytoplasm. This should start to sound a little familiar. Right, we've talked about this with muscles before. Where does the calcium come from? Well, it's kind of like skeletal muscle, in that we get it from the sarcoplasmic reticulum. But also remember the smooth muscle, we got calcium from outside the cell. Right? So the cardiac muscle cells get calcium from both the sarcoplasmic reticulum and extracellular fluid. When that calcium comes in, what do you think it binds to? Well, what do we already know about cardiac muscle cells? They're striated, okay? So it seems like striated, so it's sarcomeres, its filaments are a lot like skeletal muscle. Skeletal muscles, thin filaments have tropomyosin and troponin. So if that's the case, then calcium is probably involved in that. If not, then it's probably involved with calmodulin like smooth muscle. Well, in this case, it's like skeletal muscle, right? So that calcium that comes in binds with troponin. And then tropomyosin rolls out of the way. Myosin can bind with actin, and in the presence of ATP, you go through that sliding filament model. And you get contraction. You guys know how that works. Remember the myosin head's pulling on actin. Okay, we got that? Let's look at figures. So here's a contractile cell in the heart. Our action potential travels along the cell membrane. It can pass through the gap junction, so the cell gets the message, triggering calcium channels in the cell membrane to open, and calcium channels in the sarcoplasmic reticulum to open. Calcium diffuses into the cytoplasm, can bind to troponin, move tropomyosin, so now myosin can bind onto actin with the presence of ATP, and it can go through its cycling to pull on those thin filaments. The sarcomeres shorten, the muscle contracts. It's all review right there, for the most part. Right? There's a few differences from skeletal muscle, but it's pretty similar. All right, now what I want to do is I want to look at the actual action potentials of these cells. Now in this case, it's going to be different than a typical neuron or a skeletal muscle cell. So myocardial action potentials. Remember, myo means muscle. Cardio refers to heart. So these are heart muscle cells. Turns out, not all of them are the same. We're going to break them up into two groups. So within the myocardial cells, we have contractile myocardial cells. As you can probably guess, these are the cells that contract. And then we also have autorhythmic myocardial cells. These cells do not contract, but as the name suggests, they set the rhythm. These are pacemaker cells, just like we saw with smooth muscle cells. We talked about smooth muscle cells being autorhythmic. Some of them have pacemaker potentials. We're going to see that with the autorhythmic cells. So contractile cells and autorhythmic cells, they each have different action potentials. They're going to look different. Let's start with the contractile cells. All right, so the action potential for a contractile myocardial cell. For some reason, we're going to start with step 0. As opposed to step 1. All right, so first step, sodium channels open in the cell membrane. and the cell depolarizes. So which way is sodium going to diffuse? So we come into the cell carrying its positive charge with it. So far, normal stuff, we've seen it before, nothing new. Okay, so we had our resting membrane potential for those contractile cells. Cell gets stimulated, so sodium channels open, sodium diffuses in, and you get this rising phase of the oxygen potential. All right, step one, potassium channel is open. The cell begins to repolarize. Again, this is stuff we've already seen before. Our sodium channels will close, potassium channels are open, potassium diffuses which way? Out of the cell, carrying its positive charge with it, starting to repolarize the cell. All right, nothing new here. And we see that with this little dip, we're starting to repolarize. All right, now we have a change. After you started just a little bit of repolarization, you get a decrease in potassium permeability and an increase in calcium permeability. And that causes the plateau phase. So if you decrease potassium permeability, less potassium is leaving the cell. And if you increase calcium permeability, which way is calcium going to go? That's coming in. So that calcium coming in brings more positive into the cell. So in a way, the potassium and the calcium are sort of working against each other. Potassium's repolarizing the cell, where calcium you'd think would almost depolarize the cell. But instead, what happens is it kind of levels it out. And that's what we call the plateau phase. So you can see that the repolarization has slowed way down. Sort of drags this out, and you get this phase where you don't get much change in the membrane permeability or membrane potential. Everybody understand what the plateau phase is now? Decrease potassium permeability, increase calcium permeability. All right. After the plateau phase, calcium channels close. You increase potassium permeability again, or potassium leaves. You finish repolarizing the cell.
So is there like a point at which it's like the balance of potassium that encourages calcium the calcium channels to open up again, or to open up?
I believe the calcium channels have just a delayed opening.
Okay.
I think that's what it is, but I'm not 100% sure on that. And that's where the calcium is coming from that I mentioned earlier when it enters into the cell from extracellular fluid. It's this calcium.
Okay.
All right, so once you finish the plateau phase, calcium channels are closed, potassium channels are all opened up again, you repolarize the cell.